Thank you very much for the opportunity to speak here about this project. And I would also like to thank the foundation, really, because this project was initiated by the foundation, and we, as InnoSer as a company, had a privilege to characterize this mouse model. And also in collaboration with the team of Neurospector in Amsterdam, who really did an amazing job on the molecular phenotyping of this mouse model. So it's about a mouse model with a patient-associated mutation. So it's basically sort of a next-generation STXBP1 model compared to the one that we use, which lacks one copy. How do I go to the next? The green one, right? It doesn't work. Oh, there we go. So a little bit about the project. So it's basically an STXBP1 mouse model that has one wild-type STXBP1 copy and one with the patient-associated mutation. And our goal was really to characterize this mouse model, to really describe it, so that it can be used for potential therapeutics. Thank you. So it's a pathogenic mutation which introduces a premature stop codon, and this leads to a nonsense-mediated decay, and ultimately resulting in lowered STXBP1 protein levels. So that's relatively similar to the established STXBP1 mouse models, but it does open the door, of course, to new kinds of therapeutics, because there is actually an extra copy of STXBP1 that's there, but just mutated. So, for example, you can consider things like DNA base editing or something at the RNA level. So it does, give more, like opportunity to look at different things. And our aim was really to characterize the phenotype of this mouse model at the molecular, behavior, but also EEG level. But the goal to really identify therapeutic endpoints that can be used in future studies, hopefully. A little bit about the development of the model. So, as I mentioned, it's initiated by the foundation. And the original attempt was to establish it on a Black 6 background. But similar to the previous talk, there were some issues to establish this mouse model and create a colony. So in the second attempt, it was tried on a DIS 129 background. And that was, in the end, successful. And one thing I should mention is that the mouse model that we use in-house in the past also was created by Matthijs Verhagen, but it was created on a pure Black 6 background. So, of course, the genetic background can have a profound effect on the phenotypes by itself. So that's important to keep in mind, because sometimes I'm comparing to the original STXBP1 mouse model. So these are the phenotypic domains that we assessed. So they were inspired by the original STXBP1 models from our lab, but also others. So first we looked at the molecular level to see if they have reduced STXBP1 mRNA or protein. We also always look at the general phenotype, like for example the body weight and the activity of the mice. We also assessed cognition. So with a well-established fear conditioning assay, which is also used in a lot of STXBP1 models. It's a very consistent phenotype. We also explored motor function, which is not something we commonly do in the original STXBP1 mouse model. So that was also a little bit unexplored territory for us, although we do it in a lot of other mouse models, of course. And we also looked at EEG. So that's especially the spike wave discharges, which are really very consistent phenotype in the mouse models. And in panel B, I showed also the sort of classic timeline that we always use in the original mouse model. So we start usually with behavior at 8 weeks of age. And then when the mice are a little bit bigger and can tolerate surgery better, we implant them with the EEG head stage. And then usually the weeks after are basically centered around EEG recordings. But we can also turn it around, so we can also do EEG first and behavior later, or do the behavior at an earlier age. But I'm just showing the classic design, let's say, because that's also the one that we use in the current study. So to start off with the molecular level, let's say, So we do see, indeed, this was performed by Neurospector, so, and I think the results are quite clear, that the STXBP1 mRNA is really about 50%. So that's also consistent with what we expected. And also at the protein level, there seems to be about 50% STXBP1 protein. So that really confirms that this is indeed, like, yeah, that the mRNA is mainly yeah, cleaned up by nonsense-mediated decay. And we did not see a sex difference, so these are both males and females. They were very similar. Then we moved on to the general phenotype of the mice. So yeah, a very crude readout is, of course, the body weight, which gives a good indication of how healthy they are and so on. So we know from the original mouse model that the mutants are a little bit lighter than the wild types. And that's also what we see here, at least for the males, in the dark red line. For the females, it also seems to be the case, but it's a little bit less pronounced. But at 11 weeks of age, we also introduce a selection effect, because we exclude the females that are below 18 grams because they don't tolerate the EEG implantation very well. So we don't exactly know how the line continues after 11 weeks. But yeah, I think in general the pattern is quite consistent with the previous model. And also what's good to mention is that the males had to be individually housed because they were severe, they had severe aggression. So that's of course something you always see in mouse models with when you house males together. But here it was especially pronounced. Yeah, and that also has an impact, of course, on the other behavioral tests. So here's one of the phenotypes that we also often observe in the original STXBP1 mouse model. So this is an assay called the phenotyper, which is basically an automated home cage where you individually house them for 2.5 days. And it tracks many things, for example, the drinking and eating behavior, also how much they shelter and so on. But here I just selected the distance moved, which is, of course, a very easy to understand parameter. So that's just how much they move throughout the cage. And we see, very similar to the original STXBP1 model, that they are hyperactive, especially in the beginning. So when you put them in, they are really hyperactive in response to a novel environment. And also the nights afterwards, they seem a little bit more hyperactive, but it's more pronounced in the beginning. And then also in the open field, which is assesses a similar parameter, so also the activity of the mice in the novel environment, but in this case it's only 10 minutes. I think it tells a very similar story. So it's a very consistent phenotype. If you introduce the mouse into a new environment, they're more hyperactive. Moving on to the cognition. So as I mentioned, it's a very consistent phenotype in I think all STXBP1 mouse models, that they have impairments in the fear conditioning assay. So this is an assay in which you place the mouse in a box where it gets a mild shock, and then the next day you place it in the same box, and then it still remembers, normally speaking, that it had a shock in that environment, so it freezes more. But the STXBP1 mice had a difficulty to remember this, and this was the case for both males and females. And for us, this was very reassuring because this is, of course, one of the most consistent phenotypes of this of all mouse models, STXBP1 mouse models. So we were really happy to see that it's also present in this mouse model. So we consider this really the key behavioral assay. We also explored motor deficit. So this is not an assay that we have performed very often in the original line. So maybe it can be further improved, but at least from the current experiment that we did not see an obvious motor phenotype in this assay. I saw in the previous presentation the catwalk, which we also have but haven't really thought of doing, but it's also interesting. But at least in the poll test, we don't see a clear a motor deficit. Maybe there seems to be a trend in the females because the wild-type females climb down very quickly. But we really compare the groups, you don't really see a clear phenotype. Now moving on to EEG, which is of course a very consistent and robust phenotype in all STXBP1 mouse models. especially the spike wave discharges. So we implant electrodes in the skull above the frontal cortex and the somatosensory cortex, and also a ground electrode around the cerebellum. And also we place an extra screw just for stability of the head stage. And what was very reassuring is that they also have very similar spike wave discharges compared to the classic STXBP1 mouse model, which was also practically very advantageous for us because we have a machine learning algorithm that automatically counts them. And of course, if the phenotype looks different, then we have to modify that. But the original scripts could really pick up those spike wave discharges. And in the original mouse model, we also sometimes see spike wave discharges with very high power, and those we didn't see in the mutation model. So at least from a qualitative perspective, it seems to be similar, but maybe slightly milder. And also when we count the spike wave discharges, they seem to be, there seem to be fewer in the R122X mice compared to the mouse line that has LEX1 full copy. So, for example, in the males, you see sometimes an outlier, for example, at the top, but that's still only 6 spike wave discharges per hour. If you compare that to the plot next to it, which is from the original mouse model, we see that it's much higher. And in fact, we typically exclude mice that have like lower than 10 spike wave discharges in the original mouse model, just to have a bigger dynamic range to see therapeutic effects. But of course, if we would apply that logic to this mouse model, then you would exclude all the mice. But still, we wanted to test if with these spike wave discharges that we do observe, if they can be also reduced with an anti-epileptic drug. So for that, we used levetiracetam. But of course, it's a little bit challenging to reduce so few. And of course, there were some mice that had no spike wave discharges, so we, of course, didn't include those. So we then used a relatively high dose of levetiracetam, so 150 milligrams per kilogram, which is about 3 times higher than we used compared to the original mouse model. And we did not see a very clear reduction of spike wave discharges. So this is a within subject design. So we basically recorded the same mouse twice, and once gave it levetiracetam and once vehicle, and we randomized that. And we did not see a very clear reduction, maybe numerically a little bit, but it was not significant. And this is in contrast with the original STXBP1 mouse model, where we used an even lower dose of levetiracetam, so 50 milligrams per kilogram. And there we do see a reduction. And also one drawback of levetiracetam in mice is that it has a very short half-life of about 2 hours. So you only have a very short window in which you can count the spike wave discharges. So yeah, we probably had some challenges to count them in that only very short time window. But in the original mouse model, we also see with, for example, a drug like lamotrigine, which has a much longer half-life, that we can really see a very prolonged effect. So with benefit of hindsight, we could have also maybe picked another drug to see it, but at least under the current conditions, we could not demonstrate it with levetiracetam. Yeah, so to conclude, so we assessed, as I mentioned, these domains. And just to go through it one by one, at the molecular level, we do see indeed that the mRNA and the protein are about 50% reduced. The general phenotype is very similar, I think, to the previous STXBP1 mouse models that have been published, including ours. And the learning and memory deficits are very consistent also. Motor function, we could not really see motor deficits, at least with the assay that we used. We also looked at hind limb clasping, which is also sometimes reported, but was also not very consistent. And with EEG, the spike wave discharges do seem to be present, but they're just far fewer than in the original mouse model. And what's also important to mention is that because of the genetic background, it's very difficult to compare the models one-to-one because, sometimes the absolute values in the test can be quite different. So we cannot really plot them next to each other and compare them. And also, not all analysis is done yet. So we still, we've now recently completed the elevated plus maze, but we have not really completely looked at the data yet. We're also still going to video score the mice to look at twitch and jumps, which are also an indication of seizure-related phenotypes. And maybe there can also be more gain from the EEG analysis, for example, the power spectrum and so on. But that work is still ongoing. So to conclude also and to discuss for a discussion, so there seems to be very clear behavioral phenotypes, but the spike wave discharges are much lower. So that's, from a biological perspective, quite interesting, because then it seems that the genetic background modifies the behavior and the EEG phenotypes relatively differently, so they don't move together. And also, the model is perhaps not ideal for testing small molecules with, or molecules with a very short half-life. But for molecules with longer half-lives, it's not really a problem, because we can expand the recording time window, so we have more time to count spike wave discharges. So, for example, antiepileptics with much longer half-life, or oligonucleotides, or AAVs, this should not really be an issue, because we can just take longer to record. And also, of course, as I mentioned, the mouse model can be used for testing therapeutics that cannot be used in a mouse model that, of course, lacks a full copy. Also, something I had to think about when I saw the 129 background versus the black 6 background that we normally use, is that also in the Dravet literature, it's known that if you have a mouse model on a 129 background, at least this one, that they have no phenotype at all. But if you cross them to a black 6 background, already at the F1 background, so that's the first generation offspring, they have a moderate seizure phenotype. And if you completely backcross them to a black 6 background, they even have they have a very severe seizure phenotype, and even it can be lethal in some individuals. So this also shows that genetic background can have a profound effect. And this strategy can also be considered, of course, for this mouse model. So, for example, you can take mutant males and cross them to wild-type females of a black 6 background. So that may, of course, make the seizure phenotype worse. But of course, this has to be tested then. And I think also for many applications, the current mouse model still can be used. So with this, I would like to conclude and thank the STXBP1 Foundation, and also especially Jim, who was the main contact person from our side. And also the InnoSer team. So Dennis Bergman really did a lot of work on this project. He really led this project and did a great job. Ha Pham, who really did a lot of the data analysis. Maarten, who couldn't be here this time, but I think he will be there next time again. And the InnoSer biotechnical team, who are somewhere in this picture below. They really all had, basically some impact on this project. And also, I would really like to thank the Neurospector team, who really did an amazing job at the molecular characterization of the model. Thank you very much.
Any questions?
Thanks very much over here. The question I have is whether or not the genetic background of your new model and the original STXBP1 haploinsufficient mouse are the same, because you're comparing them here. So are they on the same background or not? And maybe I missed it and you mentioned this.
The new model is on the 129 background, or a 129 background, and the original mouse model is on the black 6 background, pure black 6. Yeah, so they're quite different. Yeah.
Great talk. Thanks so much. Just very interesting, right? I mean, if you look at other models, there is like hyperactivity, and then EEG spike wave discharges are increased. Like, and in here, it's decreased, right? So it's very, like, and it's like contrary to the novel object, like not novel object, but the novel environment hyperactivity, right? Yeah. So how do you explain that? And since this is like nonsense mediated decay, would you test like something like antisense oligonucleotides to actually prevent it and then maybe restore some of these phenotypes? And why are you testing like anti-epileptic drugs if it's not epilepsy anymore in these mice? So.
So the first question was about, sorry, can you repeat the first question?
Sorry, yeah, it's like as compared to the other STXBP1 haploinsufficiency models, this model is actually producing a contrary effect in the EEG, right?
The relationship between the EEG and hyperactivity, right?
Correct. The spike wave discharges are actually increased if you look at the other models. But here it's completely opposite. How do you explain that observation?
My interpretation would be that the hyperactivity is not necessarily caused by something that relates to seizures or seizure-related phenomena. So there seems to be a disconnect between what we see in the EEG and the hyperactivity. So that's both modulated by the genetic background, but differently.
Right. But if you look at the other models, then there is increased EEG, for example, excitability. If you look at the neurons specifically in hippocampus or other areas, right? So it's contrary to those observations, isn't it?
Yeah, so we only record from the cortex, so we indeed don't know what happens at other levels, but
right.
Yeah, I don't know.
Okay, and about the therapeutics, would you test antisense oligonucleotides then, it's a nonsense mediated decay, so the protein is not functioning anymore, right? So what therapeutic approaches would you test here? And
yeah, I think of course you can correct it at the DNA level, if that's practically possible. And perhaps also RNA editing. I don't know if this mutation is exactly fit for that, but it could also be. Of course, it's also, the mRNA is also degraded, so it still has to be there to actually be corrected. So I don't know if that's possible. And maybe read-through drugs, there's maybe even things that have not really been developed yet that could be considered in this mouse model. And sorry, the last question, the topic was?
No, that was all.
Oh, that was all. Okay, yeah.
Thank you.
Thank you. Thank you.